What if you could achieve the exact same health outcome, let's say, significant weight loss, by pushing a biological button to turn it on and by hitting the opposite button to turn it off? Sounds a bit like a biological magic trick, doesn't it?
It does sound surprising, yeah. And that's exactly the fascinating puzzle we're tackling today. We're diving deep into the seemingly contradictory roles of the GIP receptor in managing our weight.
Exactly. So for you tuning in, our mission in this deep dive is really to unpack some truly intriguing new research. We're looking at how both activating and blocking this GIP receptor can actually lead to similar benefits when it comes to weight. Think of this as your quick route to grasping some really innovative science in the fight against obesity.
And to really get to the heart of this, we'll be focusing on the distinct ways these two opposing approaches operate, where they work in the brain specifically.
Okay. So before we get to the head-scratching part about how blocking something can be a good thing, let's establish some basics. We need to talk about incretins. What exactly are they?
Right. So incretins are hormones. They come from our gut, released after we eat. The two key players for us today are GLP-1, that's glucagon-like peptide 1, and GIP, which is glucose-dependent insulinotropic polypeptide. Think of them as little messengers sent out after a meal.
Got it. Messengers after we eat. So what message are they carrying? What are they telling the body to do?
Well, their main job is blood sugar regulation and energy metabolism.
Oh, okay.
They signal the pancreas to release insulin, which helps lower blood sugar. That's what we call the incretin effect.
The incretin effect.
Now, GLP-1 does a couple of extra things, too. It also dials down glucagon release. That's another hormone that raises blood sugar.
Yeah.
And it slows down how quickly food moves out of your stomach, which is good for avoiding those big sugar spikes after eating.
Okay, so GLP-1 is the one we've heard quite a bit about, especially with weight loss, right? It seems to have this knack for sort of quieting hunger signals in the brain.
Exactly that.
Yeah.
GLP-1 acts on specific areas in the central nervous system, areas that control satiety, our feeling of fullness. So it helps reduce hunger. What's really interesting, though, is that GIP also influences appetite and how much we eat. But the precise pathways in the brain, how it does that, that's been a bit more of a mystery.
Until now, maybe?
Until now, yeah. This really sets up our central question. Just how does GIP exert its influence on appetite?
Okay, so we know that boosting the activity of these incretin receptors, that can be a good thing. We've certainly seen the success stories with GLP-1 agonists for weight loss.
Definitely.
And even bigger effects sometimes with drugs targeting both GLP-1 and GIP receptors, these polyagonists.
Precisely. Medications like semaglutide, that's a GLP-1 agonist, they've shown significant weight loss. And then tirzepatide, which hits both GLP-1 and GIP receptors, can lead to even more substantial weight reduction. And here's where the plot really thickens.
Okay.
It turns out that blocking the GIP receptor using what we call antagonists can also contribute to weight loss, especially when you combine it with GLP-1 agonists.
Wait, blocking it? That sounds completely backwards.
It does, doesn't it?
How can hitting the brakes lead to a similar result as stepping on the gas? That's where this new research comes in, I guess.
Absolutely. That's the core puzzle. A team of scientists from Helmholtz Munich, the German Center for Diabetes Research, DZD, and LMU Munich. They recently published a study. It's in Nature Metabolism.
Okay.
And it throws some much-needed light on this exact question. It was a big collaboration, involved Eli Lilly, too. And what they found is that GIP receptor agonists, the activators, and antagonists, the blockers, they actually achieve weight loss through completely different mechanisms in the brain.
Different mechanisms. So it's not just turning the same dial opposite ways. They're using entirely different circuits in the brain's control system.
That's the crucial finding, yes. The research pinpoints distinct neural pathways that are activated by these, seemingly opposing actions.
Fascinating. Okay, let's break that down. Start with the GIPR agonists. The activators. We sort of had some idea how they worked already, didn't we?
We did. Previous research had suggested GIPR agonists could have effects in the brain, separate from the GLP-1 receptor pathway.
Right.
Specifically, the thinking was they involved GABAergic neurons. These are brain cells that release GABA.
The brain's calming signal, right.
The inhibitor.
Exactly. Helps reduce neural activity. And this new study, it backs that up. It provides solid evidence that the weight-reducing, appetite-suppressing effects of GIPR agonists, they depend on having an intact functioning GIPR signal specifically within those GABAergic neurons.
So if you disrupt the signal just in those calming neurons, the agonists don't work as well. That's what they found, yeah.
Yeah.
The effect is dampened.
Okay, that makes a certain kind of sense. Activate the receptor in these specific calming neurons, and somehow that translates to eating less, losing weight. Fine. But what about the antagonists, the blockers? That mechanism was the real black box.
It really was. Before this study, it just wasn't clear how preventing GIP from binding to its receptor could possibly help with weight loss.
Yeah.
But the new research gives us a critical piece of the puzzle. Turns out, the effectiveness of GIPR antagonists, it does not rely on that same GIPR signal in the GABAergic neurons.
Not at all.
Nope, completely different route.
So they're not using that calming pathway. Where are they working then? What's their trick?
This is where it gets really, really interesting. The study discovered that the weight loss effect from the GIPR antagonists, it critically depends on the GLP-1 receptor working properly.
Wait, it depends on the other incretin system, on GLP-1.
Exactly. To get that weight loss benefit from blocking the GIP receptor, you need the GLP-1 system to be fully functional. It's somehow linked.
Wow. Okay, so that's a fundamental difference then. The activator, agonist, works independently through these GABA neurons, but the blocker, antagonist, needs the GLP-1 system online to have its effect.
Yeah.
That's quite a twist. What kind of evidence did they actually gather to show this?
They used some pretty clever techniques. First, they had these specially bred mice, transgenic mice, where the GIP receptors were specifically deactivated only in their GABAergic neurons.
So they could test the agonist without that pathway working?
Precisely.
Yeah.
See what happens. Then they also used a really sophisticated technique called single-cell RNA sequencing, seq-RNA-seq, for short, in normal wild-type mice.
Okay, single-cell sequencing. That gives you a snapshot of gene activity in individual cells, right?
Exactly, a very detailed picture. You let them see which genes were being turned on or off in individual brain cells after the mice got either the GIPR agonist or the antagonist. Helps pinpoint where in the brain these drugs are acting.
And what did that super detailed single-cell analysis reveal? Where was the action happening?
It showed something quite striking. Only the GIPR antagonists, not the agonists. Only the antagonists activated signaling pathways in the hindbrain that looked very similar to the pathways activated by the GLP-1 receptor itself.
The hindbrain. Why is that significant?
Well, the hindbrain is a really critical control center. It manages fundamental things like appetite, energy balance. Think of it as a key regulator for keeping everything in check.
I see. So while the agonists are doing their thing with those GABA neurons, maybe somewhere else,
Likely, yes.
the antagonists seem to be making their mark by sort of interacting with the GLP-1 pathway, specifically in this major appetite control hub, the hindbrain.
That's precisely what the evidence points to. As Professor Mueller, the lead researcher, put it, this work has finally solved a mystery.
Yeah, it sounds like it.
Even though both agonists and antagonists can lead to similar results, weight loss, eating less, they get there by acting at completely different sites in the brain, different roads to the same destination.
And the key difference for the antagonists, the blockers, is this reliance on the GLP-1 receptor pathway, especially in the hindbrain.
That's the crucial takeaway, yes. The GLP-1 receptor plays this pivotal role specifically for the effect of the GIPR antagonists.
Okay, so we've kind of untangled this apparent paradox. Activating the GIP receptor helps, blocking it also helps, but they achieve it via totally different brain routes. What does this actually mean, then, for the future, for how we might treat obesity down the line?
Well, these findings really open up some exciting possibilities for developing more precise, more targeted medications.
How so?
By understanding these distinct mechanisms, agonist versus antagonist pathways for the GIP system in the brain, researchers can potentially design drugs that are, more effective, maybe have fewer side effects, by selectively hitting just the right pathway.
Right, you could potentially tailor the approach.
Exactly. Imagine being able to fine-tune treatments. Maybe for one person, activating GIP is best, maybe combined with some GLP-1 action. For another, perhaps blocking GIP while boosting GLP-1 is the better route. It gives us more specific targets.
So to sort of wrap up our deep dive here, we've explored this really fascinating situation where two opposite actions on the same receptor, activating it, blocking it, can lead to a similar positive outcome, weight loss, but through surprisingly different routes in the brain. It just highlights how incredibly intricate and sometimes, frankly, counterintuitive our biological systems are when it comes to regulating something as basic as appetite and energy.
It really does. And it kind of prompts you to think, doesn't it?
Yeah.
How might this idea, dual opposing mechanisms within one system, apply elsewhere in health and medicine?
That's a good question.
Are there other places where seemingly contradictory approaches might actually hold unexpected value, maybe by targeting different downstream pathways connected to the same molecule? It definitely adds a new layer of complexity, but also potential to how we approach drug development.
Definitely food for thought. And if you listening want to delve even deeper into this whole area of incretin research, which is moving so fast,
Really is.
we definitely encourage you to look for that original study in Nature Metabolism, check out related work. It's a truly groundbreaking field right now.
Yeah.